Okay, it's 10 after. How's your day going?
Good. How was yours?
You know what? I was working long enough in one chair that I was hunching over and my back is sore. But besides that, it's going great. I just have to sort of figure out how to not do that in the future. So I noticed that y'all did the Zoom sort of response. I asked you how things are going, and like a lot of you did the thumbs up in person, and that was cool. I think we're getting towards the point of the term where there will be likely fewer questions that are administrative in nature until we get around to the midterm time, which is shoot a week from like now. But whatever. Any questions about anything administrative or something that perhaps I maybe should have mentioned and forgot to mention just now? Administrative questions of any sort? Okay, I'm going to take that as a not yet or not now, and those are the sort of things which can also be emailed, or you can show up at office hours and answer questions related to administrative things. Okay, how about subject matter questions based on perhaps the lab from yesterday or content that we looked at yesterday? Okay, so I'm going to move forward, and please interrupt me if you do have questions anywhere along the line today. And we're going to sort of start up today by jumping into about where we were yesterday, but I'm going to be emphasizing, I hope, let's get rid of that one. I'm going to be emphasizing, I hope, some of the details which we had put off to the side yesterday to focus on the big picture. And hopefully between yesterday's big picture plus then the lab, y'all saw something like F tends to be small if the null is true, but F tends to be bigger when the alternative hypothesis is true. And then eventually, I guess we have a p-value to reach our conclusion. There are some specific details in the F ratio and the calculations of it, though, that are important to go into some depth on right now, because as we drift through the rest of the term, we're going to find that some of these things that we're learning now, we're going to have to sort of tweak a little bit to deal with a fancier model. And by fancier model, I mean something like this. It's usually not just one variable, one explanatory variable that impacts the response in most experiments that we do today. Usually, if you're doing an observational study, there isn't just one variable, one X, that is used to predict Y, like in a regression context. Usually there's more than one. And we're going to really get into that in more depth starting in chapter 3. But we have to know chapter 1, the ANOVA content, well, to sort of fully appreciate that revision when we get to chapter 3. Okay, so I'm going to sort of start here. And let me just say the ANOVA table, your friend, the thing that we looked at, and I guess in the lab, you should have dealt with the handicap against, or discrimination against the handicap situation. And please let me know if I need to make this bigger for y'all to read it. But in the lab, essentially we had these 1, 2, 3, 4, 5 handicap settings, or handicap levels, if you will. In one, the person had no apparent handicap. In one, the person in this videotape appeared to have a hearing aid in. In one, the person walked in with crutches. In one, the person rolled in a wheelchair. And in one case, the amputation there wasn't really an amputation, but the apparent amputation of the arm was visible in the camera. But other than the appearance of a handicap, or perhaps no handicap, the control group, the exact same two actors played the exact same two roles with the exact same script, answering the questions identically during this purported job interview. And so if there are any differences, it's probably not because the one job applicant said a great thing, but the other job applicant said something that was a horribly bad answer. No, the answers were the same. The only difference in the situation was the appearance of a handicap, well, one of four handicaps or no apparent handicap at all. And so in this perfectly designed experiment, you're going to want to say, okay, is there a difference across the 1, 2, 3, 4, 5 means? And if there is a difference anywhere across the 5 means, it would suggest that at least one of the handicaps is giving folks an advantage or perhaps harming people during the process of applying for a job. Okay, so the ANOVA table, and we sort of finished with that, it really boils down to it is a collection of calculations that could be done and in fact are done and then are reported as part of most any research effort that you're going through that uses this method of analysis. So ANOVA, you sort of need to know it. Okay. Now let's talk about the terms in the table. So there's these sums of squares, and that's in the sums of squares column. And there's 1, 2, 3 of them. There's SS between the treatment groups. There's what's called SS error. Sometimes that's called SS within. And then there's SS total or SSY to use our textbook's notation. Those are 1, 2, 3 things that matter. Then degrees of freedom. And from that earlier stat class, you remember degrees of freedom has something to do with how much information do we have here? Okay. And let's get after our degrees of freedom in total DF. DF total, that's that 69 number here. That basically says how many data points do you have in total? Well, subtract 1 from that to get degrees of freedom. There is a degrees of freedom between the different handicap groups. And in this case, it's, again, degrees of freedom between the, or degrees of freedom associated with the 5 handicap groups. 5 groups, well, subtract 1. We always seem to, for degrees of freedom, subtract 1 from something. Well, if we got 5 groups, subtract 1 from that to get 4 degrees of freedom across the 5 groups. And then because the total is the sum of 4 and some other number, but the total being 69, degrees of freedom for error can be found via subtraction. One way that some of your Stat 218 faculty members may have said something about degrees of freedom, and I'm going to key off it right now. So for some of you, you're going to say, okay, that works. Others of you are going to say, I don't remember anything to help me with that. But yes, degrees of freedom is how much information do you have to estimate a standard deviation? Well, if we're measuring the standard deviation within the groups, we have one group mean used to get our standard deviation for each of the groups. We have only the number of data points minus the number of groups degrees of freedom remaining to estimate a variance of sorts. Okay, so let's talk about mean square. Mean square, MS, is equal to the sums of squares over the degrees of freedom within any of these 1, 2 rows, handicap and error, or treatment and error. Okay, all you need to do is to divide the mean square by the sum of squares by degrees of freedom to get the mean square. Okay, so for example, the 30 divided by 4 should give you, well, 30 over 2 is 15. 15 over 4 is like 4.5. So yeah, that works about right. It's a little bit tougher to say in your head 173 divided by 65, but you know, 180 over 60 is about 3. So the answer should be close to about 3. These mean squares are numbers which we will take a ratio of. Literally, the mean square between the treatment groups, mean square t, okay, over mean square error is equal to that F ratio. And why do we care about the F ratio? Well, it's a measure of how different our groups are relative to the spread within the groups and the number of data points from each of them. And here's a secret that is very helpful to know. So MS error, if you will, R 2.666, is an estimate of sigma squared. So there's some number out there called sigma, which is the presumed equal standard deviation across all 1, 2, 3, 4, 5 treatment groups. And if you know there's a sigma from each of those 1, 2, 3, 4, 5 treatment groups, you might want to take the standard deviation of the 5 and somehow average them. Mean square error could be thought of as the average standard deviation across the groups. But then I'm going to square it because this is called analysis of variance, not analysis of standard deviations. The MS between the treatments, okay, MST, that is your friend over here, that is equal to sigma squared if the null hypothesis is true. Now, maybe you're thinking you spelled if wrong, dude. IFF, geometry from high school, it means if and only if. So in other words, if the mean square treatment, I guess I should say the average of, or the expected value of mean square treatment and the expected value there. Okay, so these are both numbers which tend to be the same if the null is true. And so the F ratio, the ratio of these two things, which are the same if the null is true, that F ratio should be about 1 if the null hypothesis is true. So yeah, the F ratio is 1-ish if the null is true, plus or minus a little bit. And the p-value is just a way of saying how far above 1 are we? Are we so far above 1 that it's really unlikely? In this case, we have a p-value of 0.03. You'd say that's below the cutoff of 0.05. I believe that at least one of these treatment groups has a different average job qualification score than do the others. Okay, we're going to reject the null hypothesis of equality across all 1, 2, 3, 4, 5 groups, and we're going to be left with concluding that at least one of the groups differs from the others. Now, that's something that I think y'all did on the lab yesterday. Are any of you nodding? Yes, and a couple of you down there who are not showing anything could do a thumbs up or a green X, or no, it's a green check mark, red X, whatever it is. I'm assuming that you did it because that was what you were asked to do, and you're all dutiful people because you have to dot I's and cross T's to get into poly. You're probably the sort that would do that. Okay. Moving forward. Now we're going to get into something which is maybe nitpicky and technical, and those of you who like equations will say, okay, I love it. But to go with the notation our book and most other textbooks utilize, if you just tell me the number of groups, I'm going to call it K. Okay? And if you tell me the number of data points that you take from group I, I'll call that NI. N itself, I'm going to call it the sum of the NIs. Okay, so you got the total number of data points across all of the groups. And if I need to refer to an individual data point, Yij, I will say that is the jth data point from group I. So that Yij, the ij subscript really says the following. First part, which group are you from? Second part, which data point are you in that group? So if I had Y315, that could be Y from group 3, but the 15th data point in that third group. Okay, so it's just a way of keeping track of things for the sake of grabbing onto our understanding, should we need to use one of these equations later. There's y bar for group I, so like y bar 1, y bar 2, y bar 3. That sounds just like what we had done with the t-test. S1, S2, S3, those are the standard deviation of each of the group I. And because I have SI squared, I should say the standard deviation squared. There, now it's complete. And then finally, what we called the grand mean, y bar bar, yesterday, that's just adding up all the data points in each group and then add those across all of the groups and divide through by the number of data points in total. So that is the average of everybody. Caroline?
I was wondering, why do we square the standard deviation? Like, why did that happen again?
Great question.
Thanks.
Standard deviation is what we usually are feeling comfortable with. Because if I tell you the standard deviation of height of adult women in California in inches, if I tell you that number is 9, you think, okay, I'm sorry, if I tell you the standard deviation is 3, you think the typical person is within 3 inches of average. Okay, no problem there. But if I tell you the squared standard deviation is 9 and call it a variance, mathematically, it's sometimes more convenient for writing our equations in that form. So yes, a squared standard deviation, it's a variance. The method of analysis of variance basically takes the ratio of 2 variances or squared standard deviations. But those squared standard deviations are in these weird units. Like if I said that the variance of height of adult women in California is 9 squared inches, it's hard to sort of say, okay, I know what that means. So we don't tend to naturally speak variances, but for the sake of this method, we're gonna work our numbers that way. I'm going to use a layman sort of analogy here, but some of you will relate. What is pH? It's like a way of measuring how acidic or basic something is, yeah? But to be honest, pH is the log of the ratio of anions to cations in a solution. And I don't remember what that is, but why would I take a ratio of something and then take a log of it? Well, it's because it's easier to work with it in that way than the other ways that we could then utilize. I mean, if I gave you the actual ratio and didn't take the log, you could still probably work that into an equation, but a lot of the equations that you would then be working with, they would be that much more complex than you would want to look at. So we just take a log first and we use pH. In that same sense here, I'm just going to square standard deviations in this section. And you're going to say, okay, well, that's not in units I'm comfortable with, but what the hell? Let's do it. Are you good with the answer, Caroline?
I think so. Yeah.
Yeah, you know, it is a tough thing. And we're going to find later this term, there's a lot of context where the way you would naturally measure something is not the way you would want to analyze it with data. Sometimes taking logs or taking the square roots is the right way to go. You're going to see with the dioecious trees example, I guess later, the height of the tree, or DBH, diameter of branch height, is sort of related to the size of the canopy of the tree, and that's sort of a 3-dimensional thing. And the number of flowers a tree has on it, if you look at bigger trees, the flowers are always like right at the outside, yeah? And we know something like the surface area of a sphere is like the radius of the sphere cubed times some factor, okay? So like the radius cubed would probably relate to the number of flowers, or perhaps DBH cubed would relate to the number of flowers more naturally than DBH might. And so we're going to take numbers and measure them conveniently for the sake of analysis. In the case of ANOVA, we're expressing it for the sake of convenience of calculations. Okay, so happy or not, I think that's the best I can do. But I will say that the standard deviation squared or variances are not numbers that are easy to work with. Like if I told you that the standard deviation squared of error is like 2.66, you're thinking, what do I do with that? Well, I guess that means about 1.6 or 1.7 was the standard deviation. And that means the typical data point is 1.6 to 1.7 units away from the average of the group that it's in. Yeah, I don't want to have to go through that, but variance is what we're going to use for now. Okay, so here are the rules. In the ANOVA table, if you tell me the number of groups, K, I know the degrees of freedom between the treatment groups is just K minus 1. N minus 1 is the total degrees of freedom. And N minus K is the degrees of freedom associated with error. DF error is what you would often call it. And it turns out that I guess if you take K minus 1 and add N minus K, you're adding and subtracting the K. And so you're left with N minus 1. And so, yeah, we can sum in this column entitled degrees of freedom. It's like the first two things sum to the third. Boom. Same deal here with sums to squares. The sum of the first two is the third. You can sort of say, aha, 30 plus 173 is 203. So I guess I could say it that way. Add those two together to get the third. But now we're speaking in terms of equations. And let me maybe clear this and maybe bigify and emphasize some things which I think are helpful. SS error looks like, at the very far right, I'm taking the standard deviations and multiplying by the number of data points, and SS error is sort of a measure of spread within the groups. Caroline, you had your hand up. Did you want to add something or ask something?
Yeah, this might be a little bit off topic, but what is degrees of freedom telling us again? Like analysis?
How many? Yeah, so how many pieces of data do you have is degrees of freedom total. Actually, we subtract 1 from it first. How many groups do you have is degrees of freedom between the groups. Okay, so number of groups minus 1. And then degrees of freedom error, I suggested that's the number of pieces of information you have that will allow you to estimate mean square error, which is the standard deviation across all the groups squared.
Got it. Thank you.
Yeah, you know what? You say got it, and I'm thinking it's still challenging because it takes like 2 full class sessions plus extra work to grab onto this. And so you're getting part of it and continue to mull on it. Okay. By the time we hit the test, most everyone's pretty happy about this. Okay, so I'm looking at SS error, and SS error basically says take the standard deviations, square them, multiply by the number of data points in the group, add all that up across the groups. That's sort of a measure of the error in data points around their group average. SS treatment basically says how close is the group to the grand mean, the group mean to the grand mean. Square that distance, sum it, multiply by n, and that is a way of measuring how far the groups are apart from each other. So this measures group variability. And in particular, between. Now, some folks will call SS error within group variability. In fact, if you look at this form of the SS error equation, we're going to take individual data points and compare them to individual data points compared to their group mean. So that looks sort of like a standard deviation. Indeed, you can algebraically pull that guy out. Okay. I'm going to move forward a little bit because, again, we're sort of repeating some of what we did yesterday and re-emphasizing a couple of new things. Really, the set of accounting relationships within that ANOVA table basically says, take sum of squares, divide through by degrees of freedom. So S, that's in the numerator, denominator, to get mean square. And that's all true for the first and second row. Notice that we don't put a mean square for the last term. The total never happens. There actually is an interpretation of that number if I were to create it, but it is somewhat misleading and unhelpful in the context of the hypothesis test. So we just don't put it in. And our F ratio is a ratio of how much variability is there between the groups to how much variability is there within the groups. And these, again, are two numbers, which, if the null hypothesis is true, on average, they are the same. However, if the alternative hypothesis is true, the numerator tends to be larger than the denominator. So this F ratio tends to be bigger than 1 if the alternative is true. How high is it? Well, that impacts our p-value. The F distribution, if you look at that sometime in one of those tables, basically is a bit skewed, but you've got your F ratio 2.86 here, and the p-value is, what's the chance of seeing something that unusual or even more unusual if the null were true? Diana?
I was just wondering where, for the equation of MSE, on the second time it says MS error. Is that supposed to be SS error?
Ooh, yes, you are so darn right. Let's clear that and emphasize with red my mistake. Yes, thank you. SS error. You know, I'm happy to say other people who may have caught it, like in winter term of this year, didn't tell me. And maybe last spring, folks didn't tell me. And maybe even the term before that in 2022, winter term didn't tell me. So maybe you're the first one to spot it, or maybe the first one to be helpful. Either way, it's a win. Thanks. Okay. So let's move forward quickly. Actually, I'm going to skip over for now the GLM geometry that our author likes to go into. It's useful for some students, maybe about a third of students really grab onto it. I'm going to skip it for now, but you remember the Pythagorean theorem? A squared plus b squared equals c squared. Y'all remember that? Same deal. Okay. What are the assumptions or requirements for the ANOVA procedure to be realistically used? One is randomness. Either you randomly assign people to the different treatments, like in our diet and lifespan example, with the mice being randomly assigned to which diet, or the discrimination against the handicap data set where folks were randomly assigned to watch this or that or the other videotaped job interview. So we're all cool there, random assignment. Or if you have populations from which you're sampling, multiple populations, you have to independently take samples from each of them. Yay! So we can think of randomness. And that's really the nature of one whole later chapter on our book is spotting lacking randomness in your collection of data. It's like chapter 8 or 9, I forget which of the two it is, but we'll get there. Basically, you should think when someone hands you a data set, if you ask them, hey, how did you collect these data? Listen for, oh, we randomly assigned the fertilizer treatment to different plots. Or we randomly sampled frogs from these different locations. Look for randomness in their explanation. What else do you need? Well, here's one that the t-test doesn't require, but we do here in ANOVA. Equal standard deviation within each of the populations. And also normality in each of the populations. You need both of those. Okay. Now that I've told you what the book says, let me tell you some recent results that we've learned from simulations. Frankly, in some cases, even if the standard deviations aren't all the same in each population, as long as you do have normality and equal sample sizes across the groups, you're okay. Similarly, if you don't have normality, but you have a large enough sample size and equal standard deviation from each of the each of the populations, and you have the same number of data points per group, again, we're okay. But the books say you need both. So I'm going to tell you, you need both. Let's talk about how to check those with data. And I guess we got equal standard deviation and normality. And let's use the data set that you guys used yesterday, discrimination against the handicap. So I got these data pulled up and figured that out. If I've got these data on handicap and the job qualification scores, let's see if we have equal standard deviation or not. Now I'm saying check with this method called Levine, or maybe use Bartlett. Well, the way to get there, and it will feel awkward, is like we're going to use the method that we're not sure we can use until we check something based on using the method. We're going to generate some results that we'll use to then say, were those results even realistic to use? So I'm going to try to fit y by x. The score may be predicted based on the handicap. Click OK. And I'm going to go ahead and turn on the ANOVA. So actually, do I have to? Check for this unequal variances. Okay, do you see where it says unequal variances? Yeah, we've got the ANOVA procedure, right? Means, show me all the means, compare the means to each other. We'll do that one later in class today. Unequal variances, just tell me if the variances are the same or not, okay? So that test for unequal variances, there's 1, 2, 3, 4 of them that our software provide to us. Bartlett is the easiest to explain, and I don't want to take time explaining that right now. It's the easiest to explain, and so you're most likely to find the Bartlett procedure in textbooks that are a little bit deeper than ours, but not much deeper than ours. I'd say use the Levine method, and I'm going to work from analogy. I think when you go to the hardware store, you always see these products. And if you have only $10 and you can only buy one thing, what would you buy at a hardware store? I'm thinking I'm going to get one of those multi-tools, because it like can cut things, and then you can use it like pliers, and you could use it as a screwdriver. And you know, I mean, they're never the best thing for the job, right? But are they always or nearly always able to get done something close? I mean, if you have to toss one thing in your car that's going to be able to help you should something unusual happen, it's probably the multi-tool. Levine is like that multi-tool. Across all of the optimal ways of assessing whether the standard deviations are different across the different groups, O'Brien works really perfectly in one very narrow situation. Brown-Forsyth works perfectly in yet another different, somewhat narrow situation. Bartlett, it requires normality in each of the populations to assess equality of spread. Well, Levine works even if you don't have normality. And so Levine, we say, just use it. And the null hypothesis is that you have equality of spread. Your p-value here doesn't reject that null hypothesis. You can see up here nicely, this test for variances being equal. They plot the individual standard deviations, and they all look pretty much close, yeah? Because the standard deviations look close, you'd expect equal spread is not hard to believe. The null hypothesis of equal spread would not be rejected at all. Levine, use it. It's your friend. Okay, second, what about this Shapiro-Wilk procedure? Okay, well, we need normality in each of the groups, okay? The way I'm going to recommend tackling that is to generate what are called residuals and then look for normality in those residuals, because the residuals are a description of, oh, Caroline first, please.
How can we tell if something has normality? Like, what does that mean?
Yeah. Within a sample, you can never tell if the population that you sampled from was normal. We can, however, say, are our data normal enough that it could have come from a normal population? So let's get there in a second. I'm just going to show you how to calculate these things called residuals. I'll talk about what they are just a bit, and then we'll go towards exactly your question. Handicap, score, reproduce the analysis. We get our friendly ANOVA. Boom, let's create it. Means ANOVA. And under the ANOVA procedure, darn it, I see the ability to save our residuals. Now, sometimes people will say save standardized. Yeah, I'm just going to save the residuals. And you can see a new column has been created, and that really says score centered by handicap. You could replace this with a residual, if you will. This score of 1.9 was 3 units below the average score. This 2.5 was 2.4 units below the average score for that group. 3 was 1.9 units below the average score for that group. The average score for that group is 4.9, is what you're saying. Compared to the average for the group, the residual tells you how far above the average you are. And so to assess normality, rather than looking at normality in each of the samples of size 14, I mean, we got 14 data points, right? Rather than using 14 data points and then do it again and again and again and again and again to look for normality, let's lump them all together so that way they're all centered at the same spot. And let's look at the distribution of, so analyze distribution is your friend, the residuals, period. And so here we're going to see these scores that yeah, let's just stack it up. I can read this more easily when it's sort of horizontal layout. You get that by stacking, check that stack box. And I'm spotting, this looks sort of normal to me, maybe a little bit skewed. 2 approaches that you may know from the past are look at the histogram. Another one is look at the normal quantile plot. And do we see straight line here? The answer is yes, we do. And so normal is not unreasonable. We would see these residuals following a straight line if the population were normal, just fine. And we see that about here. But you also could, and this is the method that we're building towards, Caroline, you could add a normal curve. So it's just gonna, unfortunately, see, on my regular computer, it works fine. On this little tiny laptop, hard to work with. But notice how this normal curve was drawn in. We could assess then, with this procedure called a goodness of fit procedure, we can assess whether our friendly normal distribution matches the data. I can literally say, for my fitted curve, how well does it fit? How good is my fit? What's the goodness of fit measurement? And the goodness of fit procedure, I recommend Shapiro-Wilks as a procedure, okay? 0.9, I don't know what the values are that they should make sense to you, but hey, how about a p-value? 0.29, and the null hypothesis is where we've got data from a normal population. 0.29 doesn't allow us to reject the null hypothesis of normality. Shapiro-Wilks, again, sort of like that multi-tool Anderson-Darling, a little bit better, but only in certain specific situations. So I think what we're really tackling the question is the population from which we sampled normal? Well, the histogram it looks a little skewed. The normal probability plot or normal quantile plot makes me think, yeah, it could be normal. Once I add in the curve to my histogram, I could then go over and say that fitted curve, is it close enough? Is the normal curve fitting the data? Shapiro-Wilks says there's not a problem with these data relative to that assumed normal fit. Okay, Elijah, question.
Can you just go back and show how you got to the Shapiro-Wilks thing on GMP?
Yeah, it's a pain in the ass, ain't it? Okay. And this is something that I think one of the labs walks you through. but I'm not going to worry about it at the moment. I'm just going to repeat. So if you have your residuals, and I'm going to create those all over again, fit y by x. Let's create an analysis of the score as perhaps depending on the handicap. I'm going to click OK. Now I'm going to go ahead and add the ANOVA procedure, means ANOVA. Once I've added that, I can save those residuals. Boom, save the residuals. Now my residuals are called centered. We've actually created another one, but they're the exact same as before. And so these residuals, I want to look at or analyze the distribution of them. So analyze the distribution of the residuals. And let's look at that distribution. We get just a histogram at first, but if we wanted to, actually, hold on, let's unstack it. Maybe that'll be easier to work with. If we want to look at our normal quantile plot, it'll be sort of over here. And again, it is straight. So reason to doubt normal? No, we don't have one here. This straight line, which the data closely follow, is where we would expect the data values to be if life were perfectly normal. It's only a little bit up here that they seem to be a little bit below the line, meaning there are a couple more in the 2 to 3 range than I would have expected. I would have expected maybe a couple fewer and then one a little bit further out. But under our friend continuous fit, I could add a normal curve. And notice that we added the normal curve here. We drew the normal in. And now down at the bottom, or if you will, off to the very right, within that fitted normal curve, it tells you the center and the spread. All you got to do is turn on the goodness of fit. And the goodness of fit, Shapiro-Wilk says, no reason to doubt normality at all. Okay. I would like to move forward to tackle one additional thing today, if you're all okay with that. Cool. So do this and pop us back over here. And I really think a question comes to my mind, and that is, if you do reject the null hypothesis of equality of spread, I naturally think, no, sorry, I said that wrong. I really apologize for that. If you do reject the null hypothesis of, oh, what do I want this guy? If you reject the null hypothesis of equal means across the groups, you wonder which group has the mean that's different. Okay, so here's where I want to be slide-wise. So if you basically say, like you did in the lab, at least one of the handicap groups has a average job qualification score different than the others. Maybe they're all different for that matter, but if you say at least one is different, you do want to know which one is different. And in what way is it different? Is it higher or is it lower? So if the means aren't all the same, I guess I could compare the first mean to the second, the first to the third, all the way through the fourth to the fifth. And in this case, there's 10 possible comparisons. Handicap group amputee compared to wheelchair, amputee compared to hearing loss, amputee none, amputee crutch, crutch to wheelchair, crutch. So I could go through all 10. And if I simply could tell you whether mu amputee minus mu crutch is negative or whether it's positive, who stacks in as higher versus who's lower. Yeah. I mean, if I told you for each of these 10 different pairings, which were negatives and which were positives, you could reconstruct the ordering, right? So all we want to do is to estimate these differences of mean with a confidence interval. And we got 10 of them. Or in the diet and lifespan example, we have 15 of them. The problem is, if each confidence interval has a 5% rate of error, if you will, confidence intervals, they're only right, say, 95% of the time. that implies they're wrong 5% of the time. And if we've got this confidence interval, and another one, and another one, and if we got 10 confidence intervals total, and if each of them has a 5% rate of error, the overall rate of error could be a lot higher than 5%. The idea is, let's increase the level of confidence in each of the 10 we're doing. So if I go from a 95% confidence interval to a 99% confidence interval, we're talking just hypothetically here, there's a 1% rate of error instead of 5 in each of the 10 confidence intervals that I generate. And if we have only a 1% rate of error in each of the 10, the overall error rate is going to be dropping down. Maybe if we set the individual error rate at the right level, we can have the overall error rate across all of the confidence intervals to be reduced to only 5%. There's a lot of methods of doing that. I think I'll talk about Bonferroni at some point in time this term, but not now. Fisher, not now. Tukey, how about that? Oh, multi-tool. So this is the multi-tool lecture. The Tukey procedure is never the best one, yeah? But it's always one which is really close to best. And again, no matter which of the theoretically optimal ways of doing this job, Tukey will get you about the same results. And so because it gets you the same results across all of the different possible weirdnesses of life, let's just go ahead and use the Tukey procedure. Okay, so I want to show you how to do that with this diet and lifespan data. And boom. There we go. We've got our diet and lifespan data, and oh, not that one, but this guy here. So I'm going to pull this down so I'm focused on just our ANOVA right down here, right here. And you say, yeah, we believe at least one of the groups is different from the others. Maybe I'll smallify some of the things I don't really need for the moment. But if I want to compare the means across the groups, just use the Tukey HSD for honest statistical difference or honest significant difference. So that's the way to go. Again, under compare means, use Tukey. So we're turning on, it's like the little red triangle thing. We're turning on additional output. The Tukey output we have just added in. You can see that this, we got it now a set of sergeant stripes over here on the right. Ignore that for the moment. The Tukey output looks like this. It's a ordered differences report, and literally the crutch group minus the hearing loss group, that is the biggest difference. Me over here, let's look at the crutch and the hearing loss. That is the biggest difference in the means. We're saying the biggest difference between two means is 1.8. Okay, then the crutch and the amputee, second biggest difference, all the way down through the smallest difference of two means, the amputee and the hearing loss group. So order from biggest difference to smallest difference, yeah, we see our differences given to us. There's sort of like even a standard error for the difference that we could calculate and come up with a confidence interval for that difference. And this is actually not just one confidence interval, but it's a collection of 10 of them. 10 confidence intervals. You could also rely on this thing called a p-value and look for colors that aren't black. If it's in red, it says there's a noticeable difference between the crutch group and the hearing loss group. The hearing loss group has a lower, significantly lower mean than the crutch group. You can see the little red zone is, I'm in red. The rest are all in blue. None of these p-values, adjusted p-values for multiple comparisons, none of them say there is a noticeable difference. It's only the crutch group and the hearing loss group. So yesterday you did the analysis and you said at least one of the groups is different from one of the others. Now we're saying, it's the crutch group that's higher than the hearing loss group. And other than that, we don't spot any differences as statistically significant via our multiple comparisons method. You can see that in this connecting letters report. Notice that there's the crutch group, the wheelchair are done. We have the means. We order them from highest down to lowest. And in the crutch group, because it's the highest, it starts the first group, but I'll call Group A. And all of the others in Group A are not noticeably different from Group A on average. Similarly, Group B here seems to be all pairs that aren't noticeably different from each other. And because the crutch and the hearing loss group are not in one common group, one's in Group A, but not B. One's in Group B, but not A. You'd say there's a noticeable difference between these two averages. It's 5 o'clock, so I have to close with one thing, and I'm going to close with an analogy. You ever go to that party where, like, there's that one guy that you get along with, and there's that other person that you get along with, but they hate each other? A couple of you were nodding. A couple of you were smiling inside because you know that situation is awkward. You are like one of these three in the middle. You get along fine with the crutch group. You're not different from them in any way. And heck, you're not different in a noticeable way from the hearing loss group either. But the crutch group and there is clearly there's no commonality there. They are noticeably differing from each other, even though you and two of your friends are fine with either of them. You and two of your friends are fine with either of them. They are not fine with each other. So we're able to say, to quickly summarize this, the crutch group has a different average score than the hearing loss group. And besides that, we did not spot any noticeable differences. Okay, so that's it for time. I apologize that if I wanted to say one more thing, but give you one more example, but I didn't. You know what? We can stay around and chat more. I can also point you towards a videotape. I'm just saying, if you disappear, I won't be expecting anything other than that. I'm going to stop the share now. And if there are any questions, I'd be happy to tackle them before I stop the recording. But again, I'm not expecting anyone will necessarily stay for longer.
I had a question on, I don't know if my Zoom cut out or anything, but when you're checking for the equal standard deviations and using the Levine, what specifically about that number are you looking for?
Well, I'm looking for the p-value of the Levine procedure.
Okay.
And because the null hypothesis of the Levine procedure is that the populations all have the same standard deviation, a p-value above that 0.05 cutoff says there's no reason to doubt an assumption of equal standard deviations.
Okay. Okay.
So a p-value above 0.05, you're all good.
All right. Thank you.
I had a question for the Shapiro-Wilks for
Yeah.
testing the normality. You
Yep.
said that there's no reason to doubt the normality. Is that because the p-value is less than 0.5?
Oh, it was above that 0.05.
right?
Again,
Okay,
the
so-
null okay. So Shapiro, Wilk, and Levine, they feel backwards to many people at first. Because often you sort of say the null hypothesis is what you want to reject, and the alternative is what you want to conclude. The null hypothesis for both Shapiro and Levine is, oh, things are right. And so you're looking for evidence of wrongness. And if you see, oh my gosh, a lack of normality, the p-value will be below 0.05, and you say, well, I shouldn't be using the procedure. If you're looking for a lack of equality spread, if Levine is below 0.05, you'd say, oh, look, the spread is not the same across the groups. There's a noticeable difference between at least one of the groups and then the others.
Okay, so because it's greater than 0.05, we know that there's no reason to doubt normality.
Great way to phrase it. Mhm.
Oh.
Yeah, the null hypothesis, if you will, is something you like to see because that means you don't have to deal with an even more complex, problematic data set.
I'm not sure if I pulled up the data the same way, but I have a p-value that's less than 0.05 for the variances. Would that mean that
For which data set was it? Was it the discrimination
for
against the handicap?
the handicap ones? I was
Well,
wondering
I'm going to look right now
Yeah.
because I thought I thought I had seen everything okay, but I'm going to again check for unequal variance. And okay, got it. Let me share a screen here. And I think I spotted maybe what you are looking at. So so within the context of I did the ANOVA right here, and then I went up and turned on the unequal variances, right? Checking for whether the variances are the same or different. And that produced the results down here at the very bottom. And I see lots of stuff. Means, standard deviations, things like that. The tests are right here. And Levine is the one used, and the Levine p-value is 0.9396.
Okay, so I shouldn't look at the
What the heck about that guy, though?
just because it was
This
red, so
is-
I looked at it. and-
Yeah.
Okay.
Yeah. So Welsh's procedure, which we are not dealing with this term, Welsh's procedure is something they just add on for free, because if you did spot a p-value below 0.05 in Levine here, Welsh's still allows you to test whether the means are the same, even if the standard deviations are different from each other. So it's sort of like Welsh's is your backup plan. It's not ANOVA. It feels like ANOVA when you look at the equations carefully, but it isn't what you would want to use because it's less powerful than ANOVA is. You would only use it if you have to.
Okay, gotcha. Thank you.
I don't have content related questions, so if anyone has any other content related questions, go for it.
I did have one more for the homework that was due yesterday. The answer key for the last one gave the error as plus or minus 200, but I think on the homework you asked for plus or minus 50. So I was just wondering.
Ooh, okay, so I should make a change there. Yeah, in an earlier version of that homework, I used plus or minus 200, and I did not love that. And so I changed the problem to be plus or minus 50, and I did change my answer from my cut and paste. So yeah, thanks for mentioning that. If it's not fixed by tomorrow and you look and see, could you please remind me?
Yes. Thank you.
Thank you. Okay, well, I've got one person who wants to stay and chat about something, but not related to content. So the rest of you can sort of bail out.
Thank you.
Thank you.
See ya.
Okay, so I got my registrar thing figured out, but it's past the drop date now, so I think I believe I need a permission number.
Okay, so
or do I need to contact the registrar?
that sounds right. But actually, I think yesterday was the last day to add and drop officially. And so there may be a form that has to be filled out. I'd say just ask the registrar, because I think I'm looking now to see if I can spot. So yes, if I have permission numbers, it means it might work. But if I don't have permission numbers to give out at this point in time, it means you have to fill out another form. I'm not a fan, but they didn't ask me my opinion.
So just contact the registrar, and then if they need to contact you. you know.
Well, I'm looking to see if I still have permission numbers for the class, because if I do, I could just give you one and it might very well work. Oh, I do have a permission number. Haha. And out of curiosity, do you need to have any prerequisites waived to enroll, or are you just, everything's
I don't
fine?
think so. I think I was like, this is just another class I'm supposed to be taking. I don't think
Okay,
I'm.
so I am going to put into the chat just a permission number, which should work if you're allowed to add late with a permission number.
Okay.
So as far as I know, this one should work if you try to do it right now. And if it doesn't work, that means you have to ask them and see what they say. And I have no idea why
Gotcha.
we have the rules we have some days. Actually, I've talked to the registrar about a few of these, and he can always tell me a good reason for each of the rules that I think that we have that are stupid. But they do each of the rules, which are reasonable, they create problems. Because your situation is like, hello, I was just obviously waiting for something to clear and it did, and now it's too late. What? That's not cool. And the registrar is like one floor away. No, even on the same floor as the financial aid officer, the head of financial aid and the registrar. They have offices that are within 50 to 100 feet of each other. So they should be able to work this out.
Okay, it was unable to complete my request and I don't have access to perform this quote unquote transaction at this time. So I'm going to contact the registrar.
Yeah, call and if you send an email, I would say please copy me on it.
Okay.
And I will indicate, and if you get anything other than an answer of sure, no problem, here's how to do it.
Mm-hmm.
I will call the registrar himself, and unless he's in Turkey, he'll sort of try. I would ask him to, as a favor to me, make sure he could get it done.
Okay. And then, do you hold office hours tomorrow? I couldn't see it on Canvas.
Yes, it's shortly before noon. So
Okay.
like 11:25 to 11:50.
Found it.
And then
Okay.
again, 1:10 to 1:35.
Perfect. I suspect I will have questions once we start doing the homework.
Yep.
All right. Thank you. Have a good night.
Yeah, you too. Bye.
Bye.